Hello, I'm Andrew Plum. I'm a consultant radiologist at University College Hospital and a senior lecturer in medical imaging. So the project I'm going to talk about today is to use photoacoustic imaging to look at the brain. We have lots of ways of imaging the brain in medical imaging. Currently, CT and MRI are the most frequently used. These are morphological techniques that just let us look at the structure of the brain. But some newer MRI techniques let us look at how the brain is functioning. Largely, these are based on consumption of oxygen or of glucose. The problem with them is that they're limited by spatial resolution, i.e. how small a structure can we see, and temporal resolution, i.e. how fast can we generate the images. There's also a considerable limitation with MRI in that the magnet that we need to generate the image is very large and the equipment is expensive and immovable. So the patient has to come to the scanner, which may not be possible if they're very sick. It would be great if we had an alternative technique that could address some of these weaknesses. One of the possible solutions for this would be photoacoustic imaging. This is a technique that relies on illumination of the tissue of interest with laser light. Because we can adjust the wavelength of the laser, this means that we can image different components of the tissue that we're interested in. For example, oxyhemoglobin and deoxyhemoglobin absorb to different amounts at different wavelengths. This means that we can image the distribution of those two components. When the brain is using a lot of oxygen, that means it's very active, and therefore, by imaging those two components, we can work out which parts of the brain are doing the most work. To explain a little bit more about the physics of the project, I'll hand over to Brad.
Hi, my name is Bradley Treby and I am an EPSRC Early Career Fellow in the Department of Medical Physics at UCL, and I work in ultrasound. So as Andrew mentioned, photoacoustics works by irradiating the tissue with short pulses of near-infrared laser light, and those photons go into the body and then they're absorbed by different structures and generate ultrasound waves. And those ultrasound waves then propagate back out to the surface of the tissue where we record them using an ultrasound probe. So a key component in photoacoustic imaging is to take those recorded ultrasound signals and back project them or reconstruct where they came from to give us a photoacoustic image. So in conventional soft tissue photoacoustics, that's relatively easy because the tissue is quite homogeneous, so it has the same sound speed throughout the tissue. But in photoacoustic imaging in the brain, you have the skull in the way, and the skull has quite different mechanical properties to the rest of the body. So one of the key things about this project is to work out numerical models that we can use to do the photoacoustic image reconstruction. That is, to go from the measured ultrasound signals back to a picture of what generated them, so i.e. the structures in the brain. So we're going to use various new novel techniques which include meshless techniques, which mean we can get nice smooth pictures of the skull inside the model. So UCL is a fantastic place to do this project because of two reasons. First, we're a world-leading institute working on photoacoustics, including the translation of photoacoustic imaging into the clinic. Secondly, we're also a world leader on computational models of acoustics and image reconstruction, and it's those two components that are going to make this project work.